Good evening, everyone. Welcome. It's great to see you all. It's so fun to finally be back in person after a winter of Zoom meetings. I think we're all excited to see you, and it's great to have everyone here. For those of you who I don't know, my name is Jerry Bowers, and I'm the VP of External Affairs and Chief of Staff here at the MDI Biolab. And we are so excited to kick off our Summer Science Cafe series with you. We've got a great lineup planned, everything from our folks here are going to tell us a little bit about rebuilding kidneys and growing kidneys, artificial kidneys in a dish using patients' own stem cells, which is pretty exciting, to talking about the right whale issue, to talking about Alzheimer's, to talking about science communication. And then we're going to wrap up our season with a great session on art and science. So we've got something for everybody. And if you haven't taken a peek at the lineup, that's on our website at mdibl.org/events, and you can see the lineup. In addition to our science cafes, we also have our traditional summer events. So July 11th, we have Family Science Night, and we have Dr. Bob in the audience, if anybody sees. And then we have just a great lineup the week of July 22nd, which is our homecoming week. So we have lots of activities planned. Our annual meeting is on July 25th, and just a ton of activities happening this summer. So check them out, and we hope you will join us for sure. I also want to just give a shout out in the back. You see lots of student faces there. So we welcomed our 2024 undergraduate students to campus last week. So we are very excited to have them with us. And thank you all for being here this evening. So if you are interested in what it's like to be a student here at the MDI Biolab, I'm sure one of these fine folks would be willing to have that conversation with you. But for now, I am excited to turn things over to my colleague, Fred Bever, who's going to introduce our speakers for tonight's Science Cafe. I do want to just acknowledge our sponsor for the evening, Cross Insurance. They've sponsored this entire series, so we're very grateful to them for that funding. And with that, I'm going to turn it over to Fred. Thank you so much.
Thanks, Jerry. So I'm the Chief Communications Officer here, and I get the wonderful opportunity to work with people like Iain Drummond and Leif Oxburgh pretty frequently. And as a layperson, it's a real honor. I'll tell you a little bit about them, and I hope our feedback is okay. So Leif Oxburgh is a PhD, got his PhD at the Swedish University of Agricultural Science in virology, I believe. He did his postdoc at Harvard. He was a senior scientist at Maine Medical Center for several years, where maybe that's where we all first got to know Leif a little bit. He was a full professor at Tufts, and now he is the scientific director at the Rogosin Institute in New York City, which is dedicated to kidney treatments and kidney studies. Our own scientific director, Iain Drummond, got his PhD at the University of California at Berkeley. He did his postdoc at Northwestern University Medical School and the University of Chicago. He was an assistant professor at the Harvard Medical Center and Mass General Hospital, where he was also the Harvard Stem Cell Institute the director of the Harvard Stem Cell Associate Professor of the Harvard Stem Cell Institute Kidney Program. I thought you directed that.
Oh, yeah, I did.
Oh, yeah, okay, good. Where he established the zebrafish as a wonderful model for kidney studies and for human kidney genes. And now he's our scientific director at MDI Biolab. Leif, you were a visiting scientist, I know, in 2019. Were you before then as well? Because we have a photo of you from 2015, I think, in our files.
That was, I think, a meeting I was at here, a conference where I met a lot of the people who I subsequently came back to work with. So it came in stages for me.
Okay. And and when you were visiting scientists here, you worked directly with Iain. Right?
Yeah,
You were working
that's right,
together
yeah.
on these kidney studies.
Yeah.
And broadly speaking, what these gentlemen are working on is how you can take some of the regenerative capacities that the zebrafish has, and many people at MDI Biolab know that the zebrafish are really incredible fish, which Iain has qualified many of its capacities for regenerating, particularly its kidney, and taking those capacities and applying them to human stem cells so that we can create tissues that function in a regenerative way, which Leif is working on the mammalian side of that. And they're meeting in between, I think, and working together to try to really make that happen. So first, we're going to go back a little bit and just talk about the kidney and what we know about it and how we know that. And Iain is going to kick that off for us.
Okay, thanks, Fred. Nice introduction, and thank you all for coming. I hope we grab your attention. The presentation is fairly simple, and we welcome you to interrupt us with questions at any time. And now I think I have to stand up, Fred.
Please do. Should they get a walker? All right. Want to get out of the way?
No, I think we're all right, except this isn't working.
Well, did not.
Pink, pink. I'll go over here.
Ah,
All right, how about that? Okay, a brief history. I thought we'd start. This is the outline of our presentation for tonight. We'll give you a little history of the kidney, in particular about MDI-BL and how it works and what goes wrong, and the issue, the current health issue that's facing all of us, and then what we're doing to try and fix it. So diving in head first, or fish first, for the benefit of the students who may not know this institution, MDI-BL, was a founding mecca for kidney physiology in the latter half of the 1900s. And these were seminal discoveries made by people here at MDI-BL that figured out how fluid secretion could happen across epithelial tubules and the mechanisms, the ions driving it. And you can meet Patricio Silva. He's walking around here. It's maybe not here tonight. But so MDI-BL had a major role to play in how the kidney works and what we know about it. And this guy, Homer Smith, was one of them. You can go down to the waterfront down at the Star Point and see the kidney shed, and that's where this guy worked. He's responsible for really figuring out how fluid is filtered in the kidney and how the kidney actually works to maintain the osmotic and salt, sugar, amino acids, all the good things in your blood to keep all your other organs working, which is why he equates the kidney to the source of all philosophy. So it was a wonderfully broad claim for us in the field.
wait, can you say that again? How it's is the basis for philosophy?
Well, a lot of people think that the function of the kidney is to make urine, right? Well, that could be further from the truth. It's like saying the function of your air conditioner is to heat up the air outside your house, because all the waste heat and all that is discarded, right? The kidney's role is to maintain fluid homeostasis, salts, potassium, chloride, all those things, such that your heart can continue to beat and your brain can continue to function. Because as soon as all those ion concentrations go out of whack, nothing works. Okay, so the kidney is the most important organ in your body.
Amen.
And it's also the most important organ in evolution, because life evolved in the ocean. Cells grew up in a salty environment. And for an animal to get out of the ocean and get onto dry land, they had to carry that salty environment with them. And the kidney provides that regulation of body fluids such that the saltiness of your blood is maintained. The ocean's a lot saltier now than it used to be, but the hypothesis is that at the time evolution walked out of the ocean, it was about the level of your blood. So that's important. So how does it work? Well, the kidney filters blood all the time. And that just takes place in this little thing called a glomerulus up here. So there's a capillary flows in here, and by pressure-driven filtration, the plasma, not the cells and not the big proteins, but the plasma gets filtered across that and travels down this tube. And as it travels, all the physiology, the pumps, ion channels, take back all the good stuff and let stuff you don't want go past. To me, it's like those wonderful sushi mart restaurants you go to with the dishes floating past you, and you can just sort of, oh, that's a good one. No, that one looks ugly. And basically, it's a selective recovery process that allows you to maintain the blood concentrations of all the good stuff like salt and amino acids. The kidney also concentrates water. This is a loop of Henle that has a magical cross-current exchange. And the husband of Susan Fellner, who's sitting right there, came up with the explanation for how this works. And he visited here as well during his time of discovery. So basically, the kidney is filtering blood constantly. And in fact, it filters about 180 liters a day. And if you think about that, that's about the size of a shopping cart full of blood. Okay, it's kind of a gross image, I agree. But that's the amount of blood that actually passes through your kidney. And so it's an incredibly busy organ, incredibly active at this blood filtration. And I think the real question is, what goes wrong?
Which brings us to Leif, is going to tell us a little bit about what can go wrong and some of the pathways that are of investigation that are out there now that's maybe showing promise, including what Leif and Iain are working on.
Thank you. Yeah. So as you can see from what Iain's told you, this is a very busy organ. It's continuously working. It takes 20% of all of the output of your heart and processes that on a continual basis. So it's operating at a very high physiological level, and it's prone to many, many, many different kinds of disturbances, which means really that about currently maybe 14 or 15% of the entire adult population of the US has some form of chronic kidney impairment. That's all of it. If we look in this room, it's a lot of people. So this organ ages and is susceptible to toxic injuries, to injuries from when you dry out, when you undergo cardiovascular surgery. There are many, many reasons that the kidney becomes damaged as we age. And if you look at just the numbers, you can see here, if you look at the graph on the left, this is because kidney health care is regulated at the federal level. We have really good statistics for national statistics for kidney disease. And what you see to the left there is the graph between 2001 and 2021, which is the most recent data, showing how many people in the United States have end-stage kidney disease. And end-stage kidney disease is defined as the point where you can no longer support essential functions because you've lost so much kidney capacity. And the first thing that you notice from this is that there's a lot of people, 825,000 people, so close to 1 million, who have insufficient kidney function for performing daily life functions. And the second thing you notice is that the curve is on upward slope. And that really has to do with the origins, as I was saying, of the types of damage that we continuously are subjected to. And they include the major offenders when it comes to kidney injury and that long-term degradation of kidney function are high blood pressure and diabetes. And those two diseases are, if you look at them, also generally on an upward slope. So this is a sort of a knock-on effect of many other healthcare problems that we have in society that are currently growing. So our best solution to that is transplantation, because if you get a new organ, you get all of the functions of that organ back, right? And so if you look at the graphs to the right, you can see, if you just look at the blue one, what you see there is that, again, this is the most recent data, 2021 and 2011 is when this starts. This gets a bit weird over here, the curves, and this is the pandemic. The pandemic caused a lot of statistical oddities between 2019 and 2020.
So that flattening out.
Yeah, so this flattening out may or may not be real, actually. And there are underlying reasons for that, but I won't go into here. But and you can see here that there's similarly some kind of odd looking trends in the curve. But you can see here again, if from the first impression, there's an impressive numbers, 25,000 transplants in a year in the United States. And that's a very impressive number, considering we started at 0 in 1955, So this is a huge success, and no one can say that it isn't, but it absolutely does not match this number, right? So there is not enough transplantation to fit the current need of this massive need. Yeah.
Expand on that. So everyone on the left-hand side is probably on dialysis?
So a lot of people are on dialysis, yes.
100%, maybe?
I think the numbers today probably are about 450,000, 500,000 people on dialysis.
Okay, so
Yeah, and this is implantation. So this is, well, this is an annual number. So this is 25,000 a year. The transplant patient lives a lot longer than a dialysis. The dialysis patient, if they don't get to transplant, the stats say that they don't have a lot of longevity, which means that when we look at the total number of people walking around with transplants, because they live a lot longer, there may be 200,000 people, 250,000 people walking around with transplants. Whereas we have 450, 500,000 people walking around who go and dialyze 4 times a week and are hoping and are on a wait list, but it's a small number of them. There's also other problems that some of these novel technologies are going to try to address, and some of those are that not everybody is eligible to be on the transplant list, because the transplantation requires a lot of aggressive management of immunological responses, because you reject this tissue. So in order to get a transplant, you have to be able to tolerate the medication and all of the surgery. And many of these individuals who are on dialysis are maybe just too frail and have other underlying reasons that they can't be on the transplant list. So the transplant list is, probably 150,000. So it's not as bad as 5%. It's not great. It's not great. People wait for a long time for a kidney. So there just isn't enough material. And so as we just mentioned, the traditional therapies right now, most people in this treatment modality to the left, which is that they dialyze. And, it can look very much to the outside world like, kidney is a solved problem because there's dialysis, right? But as I said, it's not, there are a number of reasons you wouldn't want to dialyze. One is that it's a very invasive process. People spend several hours, several days a week doing this in dialysis centers. And there's a movement now to get home dialysis, to get people to do this at home, which is great because people can manage their own care, but that relies on having a partner who can really help you with this. And so most people dialyze still in centers like ours. And basically what we're doing here is we're replacing the function of the glomerulus that Iain talked about. So instead of having this perfect little machine inside that has this perfect filtration apparatus and this amazing ability to reclaim all of the solutes that we need, we take blood out of the patient and we run it through a filter. You know, and then we extract the toxins away that build up otherwise in the patient, and then put that conditioned blood back. And that's the basis of dialysis, and that's also why it has to be done so frequently by these patients, and it really affects quality of life. There's a lot of, you know, it's not fun, and that's why so many people want to get off that dialysis and get the transplant. And here to the right, you can see the other, obviously, the other current solution, kidney transplantation. And here you can see, you know, a brand new donor kidney being put into an individual with two kidneys that are too damaged to function properly.
Now, I'm just going to interject here for a moment. Leif mentioned the RBK project, which is a response to the scope of this problem. It's an NIH-funded program through the National Institute of the DDK. It's
Digestive diabetes and kidney.
Thank you. And so it's a $100 million project that started around the time you guys were in 2019, when you were working together. And it finances various methodologies to try to get at this problem, to try to reduce reliance on donation and dialysis, and to make various, even those processes better, I think, and also prevention. And so you can point out for us some of the main pathways that are being looked at now with this fairly big effort.
Yeah, so if we look at, you know, what examples of approaches that are being taken, I think, you know, there's a few different strategies. There's very short-term strategies, such as the one to the left, that are being implemented today that increase the access to transplant. And that's things like transplant chains. So when you have a donor, if your kidney fails and you have a close relative who wants to donate a kidney, it's fairly likely that person is not going to be a match for you. But there's going to be someone out there in the United States who is. And so a transplantation chain is run by a group who match up pairs of people who have a donor who wants to donate to you know, a loved one and someone who's close, and then they match up these large numbers of patients who all have some match in the chain, and they do. It's a logistical nightmare, I think, but they do this group sort of transplantation.
A lot of helicopters.
A lot of helicopters, and side-by-side operating rooms. So this has been, you know, this has been a way to expand access, and the best kind, or the type of transplant with the best, I think, chances of survival is the living donor transplant. So this is the best case scenario for many people, that they have a live donor. Another thing that's been really very front and center in the news, of course, recently is the pig transplantations. And this is an example of, you know, a strategy that the field is working towards. This started in the 1950s and 60s. The first reaction people had to, it's 1954, and we just did the first transplant at Brigham and Women's, was, can we do it from a primate, or can we do it from a pig? Because that would mean that there's an unlimited supply. So this work has been going on for an extremely long time. And it's really now come to fruition with genetic engineering of pigs to make the cells look much more like humans. And the avenue that we're pursuing is the avenue of taking kidney tissue from stem cells. And that really is encapsulated in this picture here, where you have an individual whose cells, body cells, you can reprogram back to stem cell state. And from that stem cell state, you can educate those cells forward to becoming kidney tissue. And the goal ultimately is to transplant this kidney tissue back into this individual. And you can see here, this loop really addresses two of the big questions. One is, can we get functional tissue? But the second is, this is my own functional tissue. So it doesn't, there's no, need to match individuals and suppress immune responses. Ideally, this is the autologous transplantation situation.
Maybe I could just, the question was about what does RBK do? RBK is not transplanting pigs, so
right.
So
yeah,
we're doing something different.
Yeah.
The Rebuild a Kidney Consortium has been around for 5 years, well, actually 10 years, but this current thing that Leif and I have been working on has been for 5 years, and we actually formulated our program here. In the summer, we were both here conspiring to apply for this. So MDI-BL has played a key role in our productivity, I think. But the goal of the Rebuild a Kidney Consortium is to both prevent progression to kidney disease as well as to generate replacement renal tissue. So there's really two prongs to it. In terms of prevention, a lot of work is being put into understanding how we respond to kidney disease and how productive repair can occur, and how non-productive repair, meaning cells that are damaged, they never come back to full function, how that contributes to an ongoing self-perpetuating kind of fibrotic degeneration. And one of our board members here, Ben Humphreys, is a prominent leader in that field of prevention and proper repair. So he'll be here this summer. And then on our side of the coin, we're the builders, and we have colleagues who, for instance, 3D print matrices and seed them with cells to try and generate proof of principle that you can make tubes, you can get them to transport, and then it becomes a scaling problem. How do you make enough tubes to handle my 180 liters of blood? So that's a real challenge. But right now, I think we're at a proof of principle stage. And what Leif will describe is our efforts to exploit pluripotent stem cells to differentiate into nephrons and try and functionally engraft those into a host. So you could imagine implanting new kidney tissue into an adult with deficient kidneys and getting that to function.
And Leif, before you jump into it, I think we should mention that your colleague and visiting scientist here, Denise Marciano, of the University of Texas Southwestern Medical Center, also was contributing to this particular project, right, under the RBK program. Yeah. And I'll also mention for people in the audience here and on Zoom that please put questions in the chat box, and we will reserve some time at the end of this so that questions come in. Sharon, do you have any immediate questions from the Zoom audience?
Yep, I have one so far. Oops, sorry. Participant says his father developed nephrosis by 1959 and was one of the first peritoneal dialysis patients in the U.S. He died in 1965. Is there now a cure or treatment for nephrosis, or is it still a problem it was 60 years ago?
I'll take a stab. I'd say no, there's no cure, but I think we know the real genetic complexity of nephrosis, basically protein leakage into the urine. Our understanding of the causes of that has advanced incredibly, and I think where the field is going now is to actually try gene therapy to try and replace damaged genes in the affected cells. So there's quite a concerted effort to take advantage of the last, I don't know, 30 years worth of genetics to try and come up with cures. But no, there is no cure that I'm aware of.
Management, yeah.
Management.
Management. Any quick hits from the audience sitting here right now? Here we go.
You mentioned that the three-step approach, make the tubes, see that they pump, and then grow them to scale. What kind of scale do you need? And could this series of steps you have on the board now with creating progenitors, but then you jump all the way to organ formation, could organoids be useful as a therapeutic tool and creating fields of or a kidney region to transplant, a zone of new nephrons rather than a whole organ?
I'll try and address your question, but then I'll give it to Leif. I think that the nice thing about the kidney is it has a unit of function called the nephron. And basically what we're trying to do is get enough nephrons functioning coherently to handle the volume. One way to at least think about the scale you need is to think about what minimum function you need to stay off dialysis. And that's about 20% of your kidney function. If you still have that, you can get by. So in terms of nephrons, that's about 200,000 nephrons. Rough, don't call me on that, but roughly. And I think that is the goal, is to try and build up organoids to a functional units.
Yes.
And he's the one who's doing that.
Yes, but there's also nothing, biology's come up with a very elegant solution, which we see in the kidney. But there's no reason to think that there aren't other shapes that would be simpler to manufacture. And one of those thoughts that's been kicking around for a very long time is that if you can make the cells, these cells that have this capacity to actually selectively reclaim the things that we want and leave the things that we don't in the effluent that goes out as urine, if you can make those cells in large enough fields, you could basically use a dialysis filter. You could build a primitive kidney with a dialysis filter on one end and then run the filtrate across that field, reclaim. And that was known as the concept was the renal assist device. And it's basically a biological addition to existing dialysis to make it more resemble the body. That may be an intermediate, and that is an intermediate that several people are still working on, to a mechanical biological hybrid device that could eventually, with advances in materials, perhaps even be implantable.
Yeah. Oh, Sharon's got.
I think about in the right size to go in and using that, which would be, I guess, artificial maybe making it that using conserve in order. Could you get like 20 mouse kidneys and try to get them all to do that job?
I think that the hope here is with these organoids, and when people look at shapes and sizes, it's there's a remarkable diversity in kidney forms between different animals. And one particularly interesting form is the form that the kidney takes in porpoise or dolphin.
Yeah.
And they look like clusters of grapes, little grapes. So it looks exactly like what you're describing. So it's like clusters of many kidneys that live together. And so that, in a way, is a natural extension for this concept of organoids. If you can grow an organoid big enough, could you make it one of this little cluster? The mouse as or the rodent as a species to transplant. I don't think anybody's put there are no good strategies to do that yet. And it's been really focused on the pig. because of the size. But they're almost a perfect match, you know, size-wise for the human. And that's why so much effort's gone into the pig.
Just to
Sharon
take
has-
this just one step, there's no real limit on where you can put a kidney, right? When you transplant in a kidney, it's actually put into your abdomen, not where it usually sits. So you could put in multiple smaller kidneys
Yeah.
perhaps to achieve greater filtration.
Sharon, you have someone from the
Yes.
Zoom audience.
This question is, do you hope to transplant this tissue directly into the sub-performing kidney?
Well, maybe this is a good point for you to sort of take us a little bit about what
Yeah.
you're doing.
Yeah, actually, we have one more question up there, but I don't know.
Okay, so in this approach of using the iPSCs, so you need to first isolate and differentiate them, and then you still expect them to grow. But once these cells are inside a patient, how do you control that there is no overgrowth of these cells inside a patient? How do you control that management, and also that they can also form a proper like clarity and functionality?
So that's a very current question, and that's pushing the limits of the current technology. So there are two answers to your question. One is that there is technology to kill cells that overproliferate. So there is a kill switch that people have built in order to be able to say, hey, you know, this tissue is not performing. These cells are, you know, because one of the, so that is an issue. And people have engineered kill switches into these cells. So that's one solution to that. But the real question you're asking, I think, is when you make this tissue, is it fully differentiated? Does it stop proliferating and achieve its differentiated state? And that question is not fully, that hasn't fully been worked out yet, I'd say. The goal is to achieve full differentiation before implanting. That's always been the goal. But I think what we're seeing is that may be very, very complex because it's very difficult to recreate. And we don't understand a lot of the environment in the juvenile mammal that conditions the kidney to the point where it just stops growing and it becomes entirely, you know, mature. So this juvenile growth phase is still an open question in the field, and that's a work in process. And then what this timeline down here that I'm sitting obscuring for you says the future. And these questions that you're raising are definitely important topics for the future. But back to this, the question that came before, if we look at this scheme, you know, how can we do any of this? And the answer is yes. If we look here to the left, these are mouse kidneys. And this is exactly what we're talking about. You know, is the goal to graft tissue into poorly functioning kidneys. And that's what this is trying to model, really. So this tissue that you can see, this white stuff that's been implanted into the kidney, this is stem cell derived, human stem cell derived kidney tissue. So these mice are profoundly immunocompromised in order to accept this tissue that's human. But when we do the experiments this way, we can find that we get a solid growth of tissue in the target kidney. And when we look at we look inside this tissue, and that's what you're seeing here to the right, you can see the host over here, which is this blue. And this is the graft. And what we're seeing in the graft here in green is the vasculature, blood vessels that penetrated all the way out. So they come from the host, they find their way out into the graft, and then they form glomeruli. And you start seeing filtration, and you start seeing tubules. So yes, we can engraft into kidneys. Yes, they become but they become extensively vascularized and achieve a blood flow, blood supply. And they do start filtering. So these are all good things. And this is exactly sort of the proof of principle that we've been looking for many years.
And to clarify, Leif, this is a human stem cell derived mouse kidney, or kidney that's being engrafted into a mouse, I mean.
Exactly. So this is human stem cell derived kidney tissue that's been grafted into a mouse in the kidney. So this is all very encouraging, but and I'm going to hand it over to Iain for this next part. But the major challenge that we really come up against is that although if we look here at the nephron, it looks like a piece of PVC plumbing in this representation intentionally, but you can see at the glomerulus end, the filter end, we find this green vasculature, blood vessels that grow into this glomerulus, and we start seeing filtration of blood, which is remarkable. And we develop the different segments of the nephron, this tubule. But the main issue with this is that this end, that normally in a normal human nephron would connect to the system of tubes that leads urine to the bladder, doesn't form. It's blind ending. It's represented here just like a club-shaped blind ending tube. And this is our current challenge that really is a roadblock to realizing the potential of this stem cell derived tissue. And this is where the fish has come to the rescue.
Fish to the rescue. So, an amazing thing about fish is they can regenerate their kidneys from injury. They can add new nephrons throughout life. And in essence, all these little nephrons form much like the organoid nephrons form as little nascent tubes, but they haven't made a connection through to the output. So this represents a alien-looking nephron with its glomerulus up here and transporting epithelium. But it's this part down here we're talking about. And then in growth or in injury, stem cells that sit at the base of the nephron proliferate, make a little aggregate, form a tube, and ultimately fuse with the output. So no fluid will flow down a nephron unless you have both ends open, because fluid is incompressible. You know, if you stop it here, it's stopped up here. But as soon as you have that opening, now you can start looking at a functioning filtering nephron. And when you think about all the things that we have goals to make in terms of replacement renal tissue, just getting flow to work, I think, is our simple today's goal. And so that's where we've been innovating and looking very closely at this junction of new and old tubes. So what you're seeing here is a new tube that's proliferating. All these red cells have undergone S phase DNA synthesis and divided as they're pushing out a tube. This represents gene expression of key factors that drive this process of invasion and interconnection of this tube with another tube horizontal here. So again, these cells are invading. They have invasive processes. The tube is proliferating outward. And at the same time, critical genes for interconnection and making these cell behaviors for driving this invasive process are being expressed. So this type of analysis is really me coming here to MDI-BL allowed me to get these pictures. So we have a really excellent microscopy core run by Frederick Bonnet. And this is a super resolution microscopy in 3 dimensions where we can make it and explore it in any way we like. So I think the key elements is that my lab can study this process in fish But it turns out all the same genes are involved in mammalian kidney development. It's just that the fish preserve that function throughout their life, whereas we stop making nephrons and stop connecting new nephrons at birth. So if we could reactivate this process in a mouse, we could get past the blind end of the nephrons that we're trying to engraft.
So Iain, so this is a zebrafish
Yes,
regenerating its kidney after injury.
right.
Okay. And it's describing various factors involved in that, and visually. But you have the data as well and the proteins, and you know what's involved. And so now you're applying that to Leif's mouse stem cells,
Right. So basically, we can isolate
the human stem cells, excuse me.
we can isolate these green cells down here that are doing the invading and interconnecting. We can get data on all the genes that are expressed in those cells. We have transcriptomes, so to speak. And so those are real clues as to what the cell is doing, what signaling pathways are active. Interestingly, they resemble, or many genes are shared between this and cancer metastasis. So cancer metastasis is an invasive process of tissue breakdown and invasion. And many of that same biological process happens here as the tubes try to connect. So there's multiple tools that we can bring to bear to create an understanding of the basic biology of this interconnection process, and then hand it back to Leif to engineer a mouse to try and engineer that process in mammals. I think that's not my last. And here's, can I hand it back to you?
Yeah, sure. Yeah, so the question is this exciting understanding. So these fish are adults. And so as an adult, a fish is able to do this on a continual basis whenever it experiences damage. And perhaps that's because the fish continues to grow throughout its life, whereas we hit stop in growth as late teenagers. So that beyond that, we're just the same. Well, we're not really the same size, but we try to be the same size.
Maintain the same size.
Yeah, but we try to maintain the same size going ahead. And so this process, like Iain says, it needs to be hot wired in us because they're very, very strong control mechanisms that ensure it doesn't happen. And when you look at the kidney, or when you look at any tube, any structure that's biological structure that's full of tubes, it obviously has to have very strict boundaries between those tubes to keep them apart. Otherwise, the organ wouldn't function if there was a bit of fusion happening here and there. And so there's very strict biological mechanisms separating tubes and making sure they don't fuse in the adult mammal. And this is what we're trying to overcome. And so although while we can recognize that many of these processes, many of these genes are expressed at some point in formation of a kidney, they're very, very suppressed in the adult. And so how do we hotwire that? How do we make it express them again? And so that is really the challenge that we've been dealing with these last couple of years is what's the delivery system that we use to get these genes to the right cells at the right time and the right place for this to happen. And that is ongoing. I can't give you the answer to that yet, but we are working with gene therapy, which is a virus-based approach, where we would take virus, we would put these genes into the virus and we would target them to the specific location where we surgically also implant the tissue. So we would do a local conditioning of the graft environment using gene therapy prior to that engraftment in order to condition that local environment. And that's really the approach that we're working on right now. We've identified the players, and we really need a good system to study this at scale. And, we've the scale part of this, being able to study 100 kidneys, 1,000 kidneys, that's a step that we haven't really been able to take because it's a very, very slow process for us to look through one kidney and look for fusion points. But with new technology that's come online and that's come online here, I think that we will be able to solve that problem quite efficiently. And I think this is and I don't know if you want to take us through this because you're
So recently, a year and a half ago, Prayag Muriwalla's lab, Marco Pende, in collaboration with scientists from Harvard University, Fabian Voigt, have implemented a mesospin microscope here. And what's terrific about this microscope is it acquires 3D images of large structures, but still maintains a very high resolution for looking at subcellular features. So this remarkable microscope, this mesospin, is going to let us, instead of analyzing sections of kidney tissue and looking for interconnections as we try to apply all this fish stuff to mammals, it'll give us the image of the entire kidney. And by looking through that digitally instead of mechanically by sectioning, we'll be able to quantify events, interconnections, after we perform various manipulations on the mouse. I think there's a lot of potential going forward for AI, machine learning based approaches to quantify digital imagery like this. And so we're hoping that this sort of needle in a haystack will become a lot more manageable as we go forward. And in September, Leif's coming back along with another collaborator, Iain Smythe, to do more kidney imaging and understand better how kidneys progress to end stage, but also how can we use this mesospin to study this interconnection event. So I think that's it for us.
And that's some early kidney imaging that Marco and his group have done.
Yeah, so Marco did that. Leif sent him a kidney that is engineered to express fluorescent proteins, and Marco cleared it, meaning made it transparent, so that you can visualize all the cell structure in a thick, not that thick, but in a kidney. So it's a remarkable achievement to avoid all this mechanical sectioning and just look at the whole thing all at once and preserve resolution. So it's a great tool, and we hope to exploit it.
Which was, if I recall, it was funded partly by, or mostly by the COBRI.
Right. So the COBRI is a center grant that has supported MDIVL for 10 years now.
And then our other supporters helped with the software that we needed for it.
And other additions to it. So we've had very generous support from donors. And so it's a real team effort to get on the cutting edge of technology, and we really appreciate the donors.
And so that's where you're headed now after all of this work that you've done so far. Just before we start to go back to questions, I do have one question, which is, how does this work align with the organoid work of or have affinities with, potential complementary with the Haller Labs work?
Well, it's very directly related in terms of vasculature. Herman Haller is innovating in the level of how do you get vasculature to develop in concert with the tubules. And without the vasculature, you don't get any function at all, right? And so it's a key step, and people really haven't worked it out yet. I think the nice images Leif showed how the host vasculature can contribute to vascularizing a donor. But there may be reasons we want the donor tissue to provide its own vasculature, especially if it gets big. If we try to aim for scale, you may want to have the vasculature in there already before you try and hook it up. So there's lots of things that need to be worked out, but Herman's has some great innovations in the vascular side of things.
And I've seen, vasculature forming in organoids in images similar to these that are being produced in the Haller Lab.
Yeah, and along with Corey.
Yeah, who's back here somewhere.
Corey, whose name I'm blocking on. What's your last name, Corey? Johnson. Thank you.
So, we started a little bit late, so I think we can go a little bit late with any questions that anyone has. There's someone on Zoom with Sharon.
Yep, he says, I understand that black bears can regenerate a functional kidney after hibernation, the time when their kidney loses its function. What has been learned from the black bear kidney that can be applied to human kidney regeneration?
That sounds like Ron Korstani's
That's
calling us up there. All right, Ron. It's a fascinating study Ron did in collaboration with Native Americans. It's not funny, it's real. To harvest kidneys after bears come out of hibernation. And the question is right, that the kidneys undergo a real torpor, it's called, when an animal goes into hibernation. And the organ function is reduced to very little, to basically snooze. So in finding that these animals wake up and rejuvenate their kidneys, Ron has uncovered some interesting molecules, but it's not complete kidney regeneration per se. It's more of a coming out of a state of torpor and into function again. So I think there's potential for some of those molecules that he's found to play out in recovery from kidney injury, but not necessarily making new kidney tissue.
Yeah, no, that's what I was going to say as well, that the kidney, when you look at the histology of those kidneys, they look incredibly damaged. They look, they look incredibly damaged. And then, a couple of months later, it's recovered. So it's that the pre-existing kidney tissue appears to recover from damage in a way that others can't. And harnessing those mechanisms to repair injury, how do you come back from an acute kidney injury when you've had chemotherapy? That kind of question would be highly applicable,
or after surgery,
or after cardiovascular surgery. Anyway, Herman.
I have a question about the program you're part of. I mean, this is NIDK, it's rebuilding a kidney, making a new kidney. It's a little bit like the Apollo program. So how many are you, and how do you collaborate, and how are you going to send the kidney to the moon?
Great food, no atmosphere. Yeah. There's about 100 people, I think, as the total team, and there are, 7 or 8 labs total.
Yeah.
10.
Yeah.
Bottom line is, when you think about this huge task, right, of making a kidney, it's so complicated if you get into it. And I think that actually inhibits our thinking. You think, well, how are you going to make a kidney? Well, we're not going to do it right away. We're going to make something that does one of the functions, and then we're going to add another function, and someone's going to discover something else that improves the function of what we've made. So it's a bootstrapping process, I think, adding on functions as we go. And when I think about how you're going to build one, I think it's kind of like an iPhone, if you think about what your phone can do, it's a phone. It's also a television, essentially. It's a camera. It's it's a computer, right? So no one invented that all at once. All the technology that goes into your phone was invented prior to the phone, and then added and compiled and improved. And so when you think of the kidney, what do you want? We always have these arguments about what do you want to do first? What's the most important thing you can make to make it a kidney? And my answer is just get the thing to filter first. And there's 2 reasons for that. One, I think we probably can do it. And 2, a major question is, are the cells mature? And, your organoids are just, developmental kidneys that really will never function. Well, one part of kidney development is that function drives form, and function drives differentiation. In other words, once you get fluid flow going down the middle of a tube, that drives maturation. There are definite changes in cells to fluid shear stress. So I think getting the flow to happen and then assessing how much did that help us in terms of maturing the cells to a really high transporting state and functional state. and then go from there. And believe me, people will come out of the woodwork with innovations, and eventually the field will synthesize it. That's my view. Yes, Jim.
So it's fascinating how this tubule is connecting to other tubules and gene expression is changing it. But what happens to the other end of the tubule, which has to connect to the glomerulus? So
You see the open proximal
when you make
tubule?
the connections distally, does that influence the gene expression at the proximal?
I'd say probably, and the reason I'm saying that is that the what happens when that so if we back up just 1 minute where Iain's answer to Herman left off was that the fluid flow plays an enormous role in maturing cells. So development, the process of the what's programmed into our cells make them develop, it takes them in terms of these epithelial cells that we're particularly interested in, takes them to a certain point where they have some rudimentary machinery, but it's really their function when they start to function that then educates them the rest of the way. And this issue is the crux of the challenge that we have, that because we haven't formed that connection, we don't know what the potential of the cells is. Number one, we do know that the glomerulus starts filtering. We do know that that end is open, but also that filtration then leads to pathological changes because there's nowhere for the fluid to go. So the cells are bound to change, all through that nephron, but probably not in a good way without having the capacity to drain at the other end. So really the answer to your question is what we want to learn. But we can say that definitely there are changes, but there may be more to do with the backflow of filtrate that we end up with that blind ending set.
So the one thing I just want to mention before we run out of time is part of this is about hope for the future. And don't want to, for people who are struggling with kidney disease, when we were speaking earlier, you used the phrase to me that everyone should have a kidney to look forward to. And the idea is really to have an abundance of tailored kidney tissue or kidneys that we won't reject. And you guys see this as a real possibility. Even though you're putting the pieces together now.
I think it'll be a long-term effort to adopt and tolerate, implanted tissue, but the technology for immunosuppression and for development of tolerance, I think that's a key step, and it plays out big time in the xenotransplants. And in the case of engineered tissue, there may be less of that rejection because it is presumably your own tissue, although there are debates about what the best cells to use for this would be. But we just have to keep working on it and show it can work. And I think when you do that, people join the parade. They contribute and get behind it. And so that's where we're at with connecting nephrons.
Yeah, I think if you see the whole, if you try and get an overview, there's a number of things that are going on right now that the stem cell, our push towards stem cell derived kidney tissue is part of a larger movement within nephrology that encompasses all stages of the process of kidney degeneration. We're also seeing new drugs come from drug companies that specifically address, degeneration in the kidney. That's been a sort of, there's been a drought really of kidney specific drug development for a very long time. And so there's been a number of enabling technologies that have come in the past, 10, 15 years that have really pushed the envelope. And so I think what we're going to see, and I think the hope for the future, is that we're going to develop therapeutics at multiple different stages here. So from the vantage point of the patient, an extra 5 years of function before getting to the point of dialysis means that's hugely meaningful, right? So, if you could have a drug that, or prevent the diabetic complications from breaking down the kidney tissue and prolong that time to dialysis, that's one big win. If you can have more supply of transplant tissue, that's another big win. If you can improve dialysis, for example, by incorporating biological portion, that's another big win. So I don't think that there's going to be one magic bullet here, but I think that from the vantage point of the individual patient, this is going to be a lot better in 10 years, the prospects, than it was just 10 years ago.
40.
Yeah.
That's really good to hear. Jerry, did you have another question?
Well, to sort of circle back and I'm curious, and if you all mentioned it, I apologize if I missed it, but Iain, can you remind me what the total amount was that NIH invested in the Rebuilding Kidney Initiative?
Oh, that's a very good question.
I think it's 100 million. That's the number I've seen a lot.
I was wondering if, as this begins to kind of wind up, I'm curious if there is any initiative among the 7 or 8 labs that you are collaborating with to continue this on. And maybe, we're seeing a growing surge in large scale philanthropic support of these sort of high risk, high reward projects like this. It just strikes me that maybe there's an opportunity there. I'm just curious.
Yeah, we have 1 year left on our funding. And unfortunately, it comes at a time when we have kind of a lineup of tools to push, to get this solved. And I think most people in the consortium feel that way. The NIH is not going to continue this collaborative consortium-based system beyond it. Instead, there are new initiatives, and we will apply. I think Leif, me, Denise, and another colleague will apply for a RC2, perhaps, which is a program grant to tackle one problem with multiple inputs. So me with the fish, Denise with cell culture, Leif with animals and transplant, and a fourth colleague looking at human interconnection in organoids. So there are opportunities, but it's never enough. In fact, most people in the consortium feel it's mostly seed money. And so there may be opportunities with industry. The NIDDK is supporting people to write grants, to partner with industry, SBIR grants. But I think they're looking for more mature technologies, and we're very much on the exploratory proof of principle end and trying to move to that validated technology with our work. So that it will require some more funding.
Initiatives for other organs out there that you're aware of, and are people looking at cardiac regeneration and other?
There's an initiative for everyone,
Yeah.
I'd say. It's been a remarkable explosion. It's been ever since the iPS cell was, Yamanaka came up with it. Everyone has figured out how to treat cells with a cocktail of growth factors to turn them into whatever. So there are beating hearts, there are little organoids of intestinal tissue. And we've had those individuals come to the RBK meetings and present, what's new in gut, And Aaron Zorn was one who came and described how they were engrafting patches of engineered new gut tissue over inflamed bowel tissue to try and basically patch the intestine. So there's a lot of innovation. And what's nice about having those interactions is you find out that many of the problems are common to all of these labs, and having everyone working on them, you can learn how to overcome, roadblocks and make progress. So it's a very collaborative field, I think. Your perception?
No, I completely agree. And I think one of the things that's really driving this field of regenerative medicine is that we are now starting to see that the promises are being delivered on. So, you know, Vertex is in clinical trials with
Islets.
a drug for juvenile diabetes that's based on stem cells. So it's basically cells, cells that produce insulin and respond to glucose. That's in clinical trials. That's currently going on. Macular degeneration is another one. You know, it's age-related degeneration of the eye, and there are several programs that are putting stem cell-derived tissue into people's eyes, both here and in Japan. And there's trials in Parkinson's disease too, with stem cell-derived dopaminergic neurons that are being injected to make people asymptomatic. So this is all, these are coming to fruition. So, from the vantage point of the pharmaceutical industry too, they're seeing the potential here is enormous. And the investment, what do you invest in? And like Iain says, every organ system has a little cottage industry. So how do we get the kidney to the front of the line?
It is the most important organ.
Yeah.
As Homer Smith laid down. So I think I'll wrap this up and thank everyone for attending here in person and on Zoom. Thank you, Leif Oxburgh and Iain Drummond for participating in this. It's been really informative and clear, which is terrific. A few items to mention. First of all, this will be online probably within the week. Sharon Linscott here is brilliant at getting it up quickly. And our next Science Cafe is in just 2 weeks. The title is What the Science Says About Endangered North Atlantic Right Whales. Well, we've invited 2 guests. One, her name is Amy Knowlton. She's from the New England Aquarium. She's been studying the endangered right whales for 40 years, often from Lubec, Maine, from a station out there. And she'll be joined, and she really knows, she's been one of the prime forces in understanding this animal and its habits and its interactions with human beings. And now these days with climate-driven change. And Nick Record from the Bigelow Laboratory for Ocean Sciences down in East Boothbay. We have a collaboration going on with them. Dr. Holler is working on screening microbiota for therapeutic effects. In this case, Nick Record, he's an ocean forecaster, a computational scientist who's been predicting and modeling what's going on in the Gulf of Maine in particular, and forage food for these animals, which has been very subject to climate change. So they'll be here in 2 weeks at 5 p.m. And then things are really getting going. Summer is coming in. The next Science Cafe will be on July 8th. It's called Sparking a Love of Science. There's some children's authors, Haley and John Rocco, who are putting out a series of books for kids about some endangered animals and some animals deserving of conservation, including the axolotl salamander. And they actually visited with us here and with Priag and James Godwin to learn more. And they'll be featured in one of these books. And they're giving a presentation around telling narratives to kids that are science-based. And so that will be on July 8th. Family Science Night is coming up on July 11th, and there'll be more. Please go to mdibl.org/events and see what's up and sign up. I just want to say thanks to Cross Insurance again for supporting the Science Cafes. And again, thanks to all of you. Really great having you.